back to my channel. Today we will dive deep into the tips to understand and score good marks for your exam, focusing on the topic cell biology, where we will discuss prokaryotic and eukaryotic cell differences and similarities for your GCSE biology. As you know, everything starts with the cell, but many students lose marks because they memorize definitions without understanding how structure links to function. Therefore, today we are going to master prokaryotic and eukaryotic cells in a way that your examiners can't catch you out. All cells have specific features like cytoplasm and a cell membrane, but not all cells are organized in the same way. Therefore, scientists have divided cells into prokaryotic and eukaryotic, and you must know those differences clearly for paper 1. Prokaryotic cells, such as bacteria, are very small and simple, but they do not have a nucleus or membrane-bounded organelles. Therefore, their genetic material floats freely in the cytoplasm as a single circular loop of DNA. Sometimes they also have small fragments of circular DNA called as plasmids. Eukaryotic cells, like animal cells and plant cells, are larger and very complex, but their DNA is safely enclosed inside the nucleus. Therefore, they can carry out specialized functions by utilizing organelles such as mitochondria, ribosome, and in plant chloroplasts. We know that mitochondria are the powerhouse of the cells, ribosomes are the protein synthesis factories, and chloroplast is the site for photosynthesis. Students often confuse bacterial cells with animal cells, but bacteria also have cell wall, flagellum, and slime capsules, which animal cells do not have. Therefore, spotting these features quickly can help you identify cell types instantly in your exam. AQA doesn't just want you to label diagram, but to explain how structure links to the function. Therefore, by the end of this session, you won't just recognize prokaryotic and eukaryotic cells. You will be able to compare them, explain them, and score full marks. Why exam pattern is important? In GCSE biology, students often know the content. They know the definitions, they know the structure, and they know the functions, but they could still lose marks in exam. Therefore, today I'm going to show you how AQA actually marks your exam answers, so you can turn your knowledge into the guaranteed exam marks. Why mark scheme matters? AQA doesn't mark exam based on how long your answer is, but on whether you hit the specific mark point. Therefore, understanding the mark scheme language is just as important as revising the topic itself. The assessment objective, we can explain them. AQA GCSE biology is marked using 3 assessment objectives, but most students only revise for memory recall. Therefore, let's break them down properly. The first assessment objective is where they reward your scientific knowledge and the key terms. You must know the scientific knowledge, proper definitions, and the proper key terms. You cannot score good marks without knowing the scientific knowledge and key terms. That's the basics of the assessment criteria. The second assessment objective tests whether you can apply that knowledge to unfamiliar situations. Therefore, you must practice using facts in context and not just memorizing them, but in an unexpected way. Let's take an example. Let's take a scenario where we can apply this knowledge of prokaryotic and eukaryotic cell, their structure and function, and learn how you can apply this to unfamiliar context. The example goes like this. Scientists have discovered a new extremophile bacterium. Thermococcus extremus is the scientific name of this bacterium, which lives in high temperature and high pressure hot spring environment. Now, this bacterium is causing issues in a newly experimental deep sea mining operation by degrading their specialized synthetic metal coated polymer pipes. Now question arises, based on your knowledge of prokaryotic and eukaryotic cell, suggest why Thermus extremus is able to survive in extreme hot environment, and propose a method to stop it from degrading those pipes without using harsh synthetic chemicals. Now here you can apply your knowledge of prokaryotic cell, especially the cell wall. Prokaryotic cell wall is normally composed of peptidoglycan, but this Thermococcus extremus, which is an archaea, most likely its cell wall is not composed of peptidoglycan. It is composed of S surface layer, which is composed of proteins rather than the peptidoglycan, which make them highly resistant to the heat. Environmental adaptation of these prokaryotic organisms often produced extracellular enzymes to break down food sources in their environment. So how can you link this structure and function and answer this question, this problem, in an unfamiliar way? The application of this knowledge would be, part one of this answer is survival. The Thermococcus extremus likely to possess a specialized cell wall, which is S layer, that is not peptidoglycan based, making it resistant to the high temperatures and pressures that would denature the peptidoglycan of the standard bacteria. The part two of the solution is the bacterium is likely to secrete an extracellular enzyme to break down the polymer pipes. To stop this without harsh chemicals, one could use biological method such as introducing a specific bacteriophage virus that infect this bacterium, or altering the pH of the water slightly to inhibit the specific enzymes that bacteria uses to degrade the metal pipe coating themselves. The second example that you can use in unfamiliar context is bioremediation, using knowledge that bacteria can digest the hydrocarbons to suggest that a novel bacterium strain was found near the oil spills might possess unique plasmid, the extrachromosomal DNA, for breaking down oil compounds. Another unfamiliar context that could be used is in medical treatments, applying the knowledge of 70S ribosomes in prokaryotes to explain why a new drug which inhibits 70S ribosome is effective against the infections while leaving human 80S ribosomes unaffected. The third assessment objective focuses on analysis, data, and practical skills. If you know how to do the analysis of the practical examples, and if you know how to read the data, and if you know the sufficient practical skills, you will be able to answer the questions accordingly and give examiners good marks material. But many students ignore this until it costs them marks. Therefore, you should always practice explaining trends, variables, and conclusions. There are two papers, each worth 100 marks, but both papers test you how you think, not just what you remember, what you recall. Therefore, every answer must be practice, relevant, and exam focused. In cell biology, prokaryotic and eukaryotic cells. Let's apply this to the cell biology, specifically to prokaryotic and eukaryotic cells. Students often say bacteria are simple, but that alone scores 0 marks. Therefore, you must use the exact scientific language that AQA expects you to use. A prokaryotic cell does not have a nucleus, but its DNA is found as a single circular loop in the cytoplasm, often with the plasmid. Therefore, naming nucleus absent and circular DNA skews the marks. Eukaryotic cells are larger and more complex, but the key difference is that their DNA is enclosed in a nucleus. Therefore, always compare cells using the clear contrast. The common exam trap that students goes into when students often list features without explaining them, but AQA award marks for linking structure with the function explanations. Therefore, don't just name mitochondria. Say They are the site for aerobic respiration, releasing energy. Command words, that is also a really important part of your examinations. Let's dive into what are the command words and how you can use them to mark full marks in your examination. If a question says state, but you write a long paragraph, you are wasting your time. Therefore, give one clear fact. If it says explain, but you only list points, you won't just access full marks. You will get some marks, but no, you won't be able to access the full marks. Therefore, link cause and effect using biological reasoning. AQA uses level-based marking for longer questions, but examiners look for structure, not storytelling. Therefore, always write in this fashion. Prove your point, write about your point, write about the evidence, then explain it. GCSE Biology isn't just about writing more, but about writing what the mark scheme wants. Therefore, if you understand how AQA marks you, understand how to win marks, you will be able to understand the scheme and understand the command words, understand the assessment criteria and the objectives. You will be able to get good marks. Thank you very much for listening. Keep watching and stay tuned with my channel. In my next video lesson, I will dive even deeper and we will discuss the past paper examination questions and we will answer them according to the AQA exam scheme, the answering scheme, and how you can get good marks and how you can ace your GCSE Biology examination. I'm Sharon Daniel. Thank you very much for listening.